Hello viewers, welcome to CEC-UGC Gurukul lectures. I am Dr. Soni Rastogi from Chemistry Department of Sri Aurobindo College, University of Delhi. Today I will be giving a lecture on pharmaceutical chemistry. The topic of my lecture is drug discovery, drug development and expensive failures. This is the second lecture of the series. It is in the continuation with the earlier lecture on drug discovery, drug development and expensive failures part 1. In today's lecture, we will talk about the stages of drug discovery and drug development like pharmacokinetics, drug design, drug metabolism and finally end with clinical trials and expensive failures. Furthermore, I will delve into the question as to why drug discovery and drug development are identified as expensive failures. Drug discovery, drug development and expensive failures part 2. The sixth stage in drug discovery and drug development is target oriented drug design. The medicinal chemists are developing drugs with 4 objectives in mind. One is to increase activity, second to reduce side effects, third to produce easy and efficient administration to the patient and for the ease of synthesis. Objective 1 is the increased activity. The activity of the drug can be increased if the drug design is selective in nature and interacts with specific targets or the enzymes present in the target to give maximum drug target interaction. Objective 2 is the reduced side effects. The side effects of the drug can be reduced if there is effective interaction between the drug and target, which results in better efficacy of the drug. Objective 3 is the easy and effective administration. The design of the drug should be such that it ensures easy and effective administration in patients. It must be ensured that the design does not lead to complexities and difficulty in delivering to the patients. Objective 4 is ease of synthesis. The route or method used for the synthesis of the drug should not be cumbersome and the procedures at the different stages of experiment should not be lengthy. Target-oriented drug design aims to modify the lead compound such that it interacts more effectively and selectively with its target in the body and thus increases the activity of the drug. Stronger the drug target interaction, higher the activity of the drug, and an increase in target selectivity will lower the side effects. Then we come to the 7th stage, pharmacokinetics and drug design in drug discovery and drug development. The compound with the best interaction is not necessarily the best drug to use in medicine. Some of the most active drug discovered in vitro shows no activity in vivo. In vitro we means in cells and tissues and in vivo we mean in animals. Pharmacokinetics drug design focuses on developing such a drug design which can overcome these barriers. 8th stage in drug design and drug development is the drug metabolism. When drugs enter the body, they are attacked by a whole range of metabolic enzymes whose role is to degrade them so that they can be excreted out from our body. If the drug is found to be polar, it will be excreted by the kidneys. Non-polar drugs are not easily excreted. So the major aim in drug metabolism is to convert the non-polar drugs into polar drugs so that they can be excreted out through kidney. The objective of drug metabolism is to convert non-polar compounds into polar compounds which can be easily excreted. Besides that, certain enzymes, say for example in liver, cytochrome P450 can carry out oxidation in drugs and are able to add polar group, hydroxyl group, which can be excreted out. So, this is the enzyme which adds polar group hydroxyl and converts non-polar drugs into polar drugs. Then comes the ninth stage in drug discovery and drug development, that is drug toxicity. It is an important step in drug development and drug design. Drugs should be tested on animals and finally on humans to see what metabolites or the end products are formed in drug metabolism. It is important to ensure that no toxic metabolites or the end products are formed. The end products in drug metabolism should be inactive or dormant in nature and removed from the body easily. It has been found a drug which is active in vivo, that is in animal, is inactive in vitro. The metabolites or the end products formed in drug metabolism should not be toxic and should be dormant in nature. The toxic metabolism can cause severe side effects and can even result in death of the patient. So, we have to be very careful that all the metabolites which are toxic or even non-toxic, they should be removed from our body. Pharmacologists face a new problem as during the development of a drug, a difference in activity of the drug in vivo, that is in animals, and in vitro in isolated cells and tissues, could not be anticipated. For example, in early 50s, this is a very good example, thalidomide was given to pregnant women to relieve nausea. Later, it was found that this drug thalidomide was taken off from the market as it caused fetal deformities. Minor impurities are bound to be present arising from the synthetic route, because synthesis always lead to certain impurities which can be neglected, and there is a certain percentage of the impurities that can be present which is very minimum or almost negligible. The toxicity effect arising from a drug synthesized by one route is not the same for the drug prepared by another synthetic route. There are so many variables involved in drug interaction in the body that it is impossible to anticipate them all. One must consider that drug will be metabolized to other compounds or end products which have their own range of side effects. It is impossible to predict whether a potential drug will be safe in vitro alone or not. Here lies the importance of animal testing. So, we can say how important animal testing is before the drug comes into the market and prescribed by the doctors. Only animal test can test for the unexpected unless we are prepared to volunteer ourselves as guinea pigs. Though unfortunate, but experimenting on animals will always be an important part of drug discovery and drug development. Then the 10th is the clinical trials. Clinical trials involves testing the drug on volunteers and patients, and it involves 4 different phases. Phase 1, it is given to healthy volunteers to test the drug potency, pharmacokinetics and side effects. Drug potency is the amount of drug required to produce a defined effect. Pharmacokinetics are the barriers that the drug faces to reach the target in the body. Phase 2, it is tested on small group of patients to see if it has any side effects and to find out what dose level should be given or what is the dosage of the medicine that should be given to the patients. It is given in phase 2 to the group consists of 30 to 40 patients to know the correct amount of dosage to be administered and to know the side effects. previously not detected in healthy volunteers during phase 1. Then is the phase 3 of the clinical trials. The drug is tested on a larger sample of patients and compared with other available treatments. Preparation which has no side effects means the drugs which it is compared with the drugs which have no side effects. This establishes whether the drug is effective or not. It also helps in finding out the right dosage. Any side effects not previously detected may be picked up with a large sample of patients. If the drug succeeds in passing phase 3, it can be licensed and marketed. Now, the last phase is the phase 4. The drug is now placed in the market and doctors prescribe them to their patients. Now, everybody in the all kind of patients can take this drug because it is now has undergone all the clinical trials and is prescribed by the doctor. However, the drug is still monitored for its effectiveness and for unexpected side effects. This phase is a never-ending process since unexpected side effects may crop up many years after the production of the drug. For instance, this is a very good example, the beta blocker Practolol used for lowering blood pressure had to be withdrawn after several years of the use since some patients suffered blindness and even death. Now, this is the structure of Practolol, and this Practolol is a beta blocker and anti-hypertension drug which was taken off from the market because it caused blindness to the patients. Then comes the patents, the 11th stage in drug discovery and drug development. Having spent enormous amounts of time and money on research and development, a pharmaceutical company wants to recoup its cost and reap the benefits of its hard work. The company needs the right to sell and manufacture its product for a reasonable period and price which recoup its cost and generate profits. Patents allow the company to give these rights, and these rights are given by government agency which give them right to market that drug. Then we come to the expensive failures, that why drug discovery and drug development leads to expensive failures when so much of money is pumped in it. Drug discovery is time consuming, risky, tedious and expensive as it involves huge investments of funds by pharmaceutical company. It takes almost $100 million to $1 billion to launch a new drug, and the time taken is approximately 12 years. So we can say that it is expensive and time consuming. The only way to lower the cost and time in drug discovery is to reduce the failure of procedures at different stages of experiment. An analysis in the field of drug discovery and development shows that the hundreds of drugs comprising new molecular entity NME are tested, but only few of them become marketed product. This is the data obtained from the drug approved from Central Drug Standard Control Organization India, and we can say that in the year 2018 number of drugs approved was 6, in 2019 number of drugs approved was 7, and in 2020 number of drugs approved was just 9. So we can see the percentage of number of drugs approved every year are almost less than 10%, which is not a good figure. From this data we can say that every year 100 of drugs undergo clinical trials, but only few of them are approved. It is extremely discouraging and de-incentivizing for the pharmaceutical companies and the scientists, as there is not much progression in the number of drugs approved. As an answer to this is that as the cost of research and development is very high, more money has to be pumped in for carrying out new projects without any money crunch. Secondly, the education policies are such that the universities, students in the field of biosciences are working on high budget drug development projects. However, they are not using the advanced technologies like high throughput screening process (HTS) for the research. HTS or high throughout screening process is an automated process that can rapidly identify the lead compound, that is the drug to be formed, and provide starting point in a drug design. Thirdly, the supply of approved drugs has gone down as the functioning of the research institutions involved in medicinal chemistry has decreased due to dwindling economies. Because of the bad economy, the supply of the fund in the R&D of different pharmaceutical company has gone down. As a result, there are layoffs and the young scientists are compelled to leave their jobs and work on other scientific researches not related to drug discovery and development. One of the ways to reduce the expensive failures is by ensuring that the researchers and scientists are aware of the advancements like genomics and upcoming technologies like HTS and making use of them for drug discovery and its development. Now we come to the computer aided drug design, that is CAD. This is a very new technique, and this is an important technique which is used in drug design just to lower the expensive failures. And the process of drug design is challenging, time consuming, costly, and involves expensive failures. What we have seen all this in our earlier slides. So recent years have witnessed the development of new computer techniques like computer aided drug design (CAD) that have helped in speeding up of drug design and reducing its expensive failures. We will talk of CAD in our further slides. It makes use of molecular mechanics, which identifies the conformation of small molecules and models the conformational changes in the biological target. This results in maximum interaction between the molecule and the target. The different approaches used under computer aided drug design or CAD are target with known 3D structure and target with unknown 3D structure. So we will deal with both of them in the next slide. Target with known 3D structure, that is the target molecule, their 3D structure is known and we design the drug accordingly, according to the target, because we know the 3D structure. It begins with the 3D structure of the target molecule being identified by NMR spectroscopy or X-ray crystallography. High throughput screening HTS is used for further identification of the target molecule and data analysis. This helps in the recognition of the probable drug molecule. The data base collected from leads to the formation of a drug bank where the potential drugs are collected. This is a very new technique which is used in drug design and drug development. Finally, the structure of the drug is modified using CAD to ensure effective and selective interaction with the target. Stronger the drug target interaction, higher is the biological potency of the drug. Whether the drug is synthetically designed or it is used by the CAD. Designing is done by the CAD, but the main concept of drug design is that stronger the drug target interaction, better is the drug in the means of higher the biological potency and lesser are the side effects. Target with unknown 3D structure. Here the structure of the target is not known, and then we know the target and design the drug accordingly. Biologically, screening is carried out to identify the potential molecules which can be probable potential drug. This is followed by drug identification through docking. Docking is the identification of receptor for a given ligand molecule among large number of receptors. Furthermore, evaluation is carried out wherein assesses binding affinity between the drug and the target through docking. The knowledge of CAD looks into the therapeutic activity of the drug on the molecular basis, and finally the 3D structure of the drug is evaluated through CAD techniques. Even in the CAD technique for the designing of the drug, the therapeutic activity of the drug has to be taken in the consideration, because the main aim of drug design is that drug should be such which should be able to cure the disease or the ailment. However, it must be noted that in practice the computational chemistry, that is the CAD, is not accurate. As a result, the design and synthesis must be carried out several times before a potential drug is designed. So then also the difference is that while we are using the CAD technique, it reduces certain number of years in drug designing, because in drug designing approximately 12 years are taken, and it lessens the cost also. But we have to design and synthesis must be carried out several times to see the potential of the drug and to see its efficacy, to see its effectiveness. An example of a drug designed through CAD is carbonic anhydrase inhibitor, dorzolamide, and another example is imatinib. These are all drugs which are designed by computer aided drug design, and these drugs, the advantage of designing the drug is by the known 3D structure of target molecule or unknown 3D structure of target molecule is that they take less number of years in their designing or less number of time. Then we come to the conclusion. Drug discovery and development is a continuously evolving process. Initially, it started with the isolation and purification of active compounds from natural products. Now, it is a long process involving various stages. Today, the research in pharmaceutical industries begins with identifying a suitable target and designing a drug to interact with the target, and finally ends with the clinical trials. Today, the process of drug discovery and development is expensive and time consuming. Drug discovery and development field witnesses expensive failures due to low approval rate by FDA, that is Food and Drug Administration. FDA, as we know, is the responsible for protecting the public health by ensuring safety and efficacy in drugs. Though research and development are substantially in progress, but the number of drugs approved by the FDA is not very high, leading to drug development being recognized as a field of expensive failures. Thank you.